Happy Tuesday, everybody. We're back once again, finishing up our chapter for this week. Getting right into the do now, it says compare the genetic languages in fish, mushrooms, and bacteria. So what do we know about the languages of genetics in these three organisms? Well, as we've talked before, hopefully you remember, DNA is always the same. Doesn't matter what species you're in, your DNA is always the same. So I'm going to write that right here. DNA is the same. That's all you got to know. And what is the language of DNA? That's A, T, C, and G. Every living thing has some combination of those four bases making up the instructions of life. Now, would this allow us to do anything cool in the lab? Maybe the answer is no, and we can just stop right here. Probably not, though, right? We should probably keep going. Oh, look, a weird, minimally labeled vial of insulin with a syringe just sitting around. That's not questionable at all. Normally, we'd avoid something like that, but today I'm asking you where this comes from. We know that the pancreas makes insulin to help control blood sugar, but how does it get into this little vial for someone with diabetes who needs to inject it? What do you think? How can we get a chemical out of someone's body and into this little glass jar? It's a little challenging of a procedure, but if you take a big enough needle and angle it just right into a patient's abdomen, we can aim it directly around the other organs and into the pancreas. Then the insulin is extracted and it can be bottled for sale. Now, we need to have a consistent source of this stuff, so usually doctors just keep some kind of running list of patients that no one will miss, and then we finally realize that I'm making all this up, and please, please, please don't think I'm serious in any of this description. In this much more realistic picture, the vial right here says recombinant DNA, and that's what we're going to be talking about today, and it's a lot better of an option than unspeakable medical procedures that, again, please know I was completely not serious about that. We don't do that to get insulin. There's a much easier, much more moral way to get this stuff. And as you can see, this is going to be an example of what we call biotechnology. Because all living things have the same language of DNA, we can take genes out of one organism and put them into others. We can remove a gene from spiders that lets them make silk in their webs, put that gene into goats, and the goat's milk will have the silk in it. Not like in giant pieces, but there's small molecules of it that can be filtered and taken out and then put back in to make this thread of silk. If that sounds weird to you, it should, but it works and it's pretty cool. And besides that, now we can sing spider goat, does whatever goats do. I don't even know, but it also makes webs. This same kind of thing is seen all over the place. You probably don't know about it, but it happens. Some fish that live in cold water have genes to protect them from low temperatures, and by putting that gene into plants, we can grow cold-resistant fruits and veggies, like these tomatoes that grow all year round in really cold climates. The mice at the bottom are not photoshopped. They really do glow like that. They look green, other colors, and different kinds of light because it's really easy to take a light up gene out of jellyfish and put it into anything else that we want. You can do this to any different organism. This is a really widely used tool in biotechnology in different lab situations, and it's pretty neat to be able to pull off. Biotech is starting to look a lot cooler, right? Once more, since DNA is universal, every living thing on planet Earth has the exact same language used to give instructions. There's no such thing as human DNA or plant DNA. It's all the same language of letters, just in different organizations, different arrangements to give us different instructions for making different living things. When we take DNA from one place or organism and put it into something else, we call it recombinant DNA, since we're combining two different sources of genes. So in this picture here, we can take the gene for something from the sample of DNA up here. You see the little kind of bluish segment there that's highlighted. Let's say that's the insulin gene. So this is a human DNA sample. We take the gene for insulin and we cut it out. So there it is right there. We cut up this little piece of DNA, remove it from the big sample of DNA, and then we can actually insert that one little gene into our bacterial DNA over here. We have this little ring of DNA from bacteria. We put that one gene we want into the bacterial DNA, and now we can take that DNA ring, put it into some bacteria, and really, really quickly make millions and millions and millions of bacteria. We then just have giant vats, whole rooms with these giant tanks of bacteria, and just let them grow and grow and grow and make us a lot of insulin really, really quickly that someone can bottle up and sell to people that need it. So there's an industry behind this. That's economic factor driving the stuff, but it's the science that lets it happen, and it gets to be really pretty cool to look at. How are we supposed to get DNA out of one thing and put it into something else? To do that, we can use what's called a restriction enzyme. We know that enzymes are types of proteins that do specific jobs from early in the year, and what these enzymes do is cut DNA. That's it. That's their job, is to act like little scissors in the cell and cut DNA. But the important thing is they only cut it in very, very specific places. So for example, this enzyme at the bottom left there called TAC1 will look all over the DNA in a cell, and anywhere that it finds these letters, TCGA, in that exact order, it's going to cut them right here where the arrow's pointed between the C and the T. Right there. It'll cut right in the middle of the DNA sequence. And this other enzyme on the right, HEY3, it'll look same thing through the entire DNA molecule, and anywhere it happens to find a G, a C, and a C in that order, those 4 letters exactly, no matter what else is around them, anywhere it finds them, it'll cut right down the middle between the second G and the first C. It's going to slice it right there. It'll snip right in the middle there, wherever that marker is, and that's its only job. Now, before I put the last bit of notes in this slide and in your paper, I'm telling you now we're going to skip a lot of things today. I'm going to combine the last 2 lessons in this chapter, so what would have been lessons 4 and 5. I'm going to combine them together and do them both right now so that we can get to a virtual lab on Thursday, because I think that'd be really cool to see. I think it'd be a lot more fun than just breaking up a little bit more. That being said, don't write this stuff down right here. I just quick want you to circle where it says recognition sequences. Just circle it on your paper, make a little star, because I think that's an important term to know. This is a recognition sequence. It's literally the sequence of letters that is recognized. It's just what the specific enzyme looks for. So TAC1 looks for these 4 letters to recognize those. HEY3 looks for these 4 letters to recognize those. And that's going to cut them in whatever place. You can see the first enzyme cuts between the first and second letter. This other one cuts between the middle 2 letters. Every enzyme is different. But just make sure you make a little note of that for yourself, and then we can cross off the rest of your notes on that page. And we're going to do a lot more of that in the next couple of slides. Just chop and get rid of a bunch of stuff we would have otherwise gone over. Right now we're going to look at 3 enzymes, 2 of which we just saw, and 1 new one, and see how they actually cut these DNA sequences. So I gave you the TAC1 here with the TCGA we just saw, HEY3 up top with that same GGCC, and now we have a third one, PST1. It's going to look for CTG, CAG, and cut between the A and the G anywhere it finds those sequences. So let's check our DNA sequences and see what we can find here. Up top, we're looking at HEY3. So we're looking for 2 Gs and 2 Cs in that order only. And then we're going to cut between the G and the C in the middle. So let's just scroll through here and scan and see what we can find. GGCC. So I see right here, I see a CCGG, but that's the reverse of what we're looking for. It's backwards, so it doesn't count. We're looking for only GGCC. So I'm going to forget about that. Keep going to the right. And right here in the middle, I see GGCC. So at that point, we're going to look at our picture, and we see right here between the G and the C in the middle is going to cut it. So I'm just going to draw a line right here. GGCC, that's where that would cut. You can leave a little cross. I'm going to just actually erase the underline that I had put in. That's right where it's going to cut this DNA. So that enzyme will snip it right there between the G and the C if it happens to find that sequence. And I'm going to keep going and see if anything else shows up, but I do not see any more G's and C's. So it's only that one place that that enzyme would actually cut it. So first question says, how many cuts did HEY3 make? And that's going to be just the one. How many DNA segments were produced? And we can now see 2. It cut this one piece right in the middle here. So you have one piece over here and another piece over here. Those 2 pieces just separate like you're cutting anything in half. Down the bottom, TAC1. It says use TAC1 to count the following sequence. We're going to look for the same thing, TCGA. So again, let's just look through. I see some T's with T's, T's with G's. How about right here, TCGA, right there. So at that point, I'm going to cut between the T and the C. So I'm going to draw my line right here between the T and the C. So we'll cut that DNA segment right there. Let's see if there's any more. TCGA I'm looking for. I see TCAG, but again, it doesn't quite fit the pattern. We're looking for TCGA, so that doesn't count either. And once again, we just see the one cut site right there to make 2 pieces. And our last one, CTGCAG, we're looking for. So a little bit bigger of a sequence. Usually you see it like this, about 6 letters. The 4 are a little bit smaller. But let's see, CTGCAG. I see CTGC. Yep, right there, CTGCAG. And this time, the enzyme is going to cut it all the way on the right side of the sequence between the A and the G. Sometimes you see it more kind of dead center like the HEY3 does up here. Sometimes you see it sort of over to one side. Sometimes you see it sort of over to the other side. So I'm going to cut it right here between the A and the G. Erase my underline. Let's just see if I have any others here. CTG, I do not seem to have any other cut sites. So each of these only cut in one place to make our DNA 2 little pieces, and that's pretty typical of what we'll say. But let's go on and see if anything changes. Once again, we're going to start with the middle of the page here where it says the middle of the DNA ladder, and we're just going to cross that entire thing off. You got some nice diagrams at the bottom if you want to keep them for reference, but don't feel the need to. I think this one at the bottom is a little bit more helpful, just for showing how things get kind of stuck together. But otherwise, we're done with that, and we're going to move on to the same thing a little bit more. We're also going to skip this section of notes, which I personally think is kind of a shame because I think I have a pretty decent barbecue joke here at the bottom of the page. I'm going to leave that on there, but otherwise, we're skipping that. So let's cross that off and on to the next part. And once again, this is just the notes version of that last picture I showed you with the barbecue stuff. So I'm going to cross this entire section off. At the bottom of the page here, just notice I gave you a larger diagram of some of the recognition sequences from before. So that's good to have for reference. We'll use that again. I'll show it to you again, though. It'll be in your notes in the following slides. But that's where we are for now. Eliminate all the animations on this so you see all these pictures at one time and kind of get a big sense of everything. I'm just going to show you how this works. So this is the basic idea of how restriction enzymes are used. Let's say we have these two molecules of DNA up here in the top left. So we've got a human gene X, so that's the sequence of a human DNA. I'll change my ink color so you can see that a little bit better. Let's go to still the red, I guess. We've got our human DNA, and there's a gene X in the middle, and then we have this segment of bacteria DNA also. We want to take that gene X and put it into the bacteria. So we want to combine them in some way to study gene X or make more of gene X, whatever we want to do. We want to be able to do it pretty fast. We don't have to want to wait for human cells to make this stuff. And it's tough to use animal cells for that. It's maybe not as ethical. So we want to put it in some bacteria very quickly. So in this example, we have two different enzymes we can use to cut the DNA. And each one cuts it a little bit differently. We see enzyme here. If you look, it has kind of a little more jagged shape to it. It's a little bit of a sharper angle in that cut with these scissors. And then we have enzyme 2. If you look, has more of a rounder shape. It's a little more of like a wavy look to it. So they're going to cut the paper, in this case genes, two different ways. We can use either one of them to cut the DNA as long as we can find that correct cut site within the DNA sequence. So we'll assume it's there. In the picture on the bottom left, I've used these enzymes to cut. So I cut the gene X from the human with both enzyme 1, so we see this little jagged cut right there, and enzyme 2, so we see that much more rounded looping cut. And then on the bottom, I cut the bacterial DNA with just the enzyme 2 for that more rounded cut. Does this work now if we try to put these things together? How would this work? We cut our two pieces of DNA, and let's see if we can actually reassemble them. So we're going to take human gene X that was cut with enzyme 1 and try to attach it to bacteria cut with enzyme 2 in this kind of middle picture here. What do you think from the look of the papers? Will these pieces match up properly? Can we fit these things together very well? Doesn't really look like it, right? I see the bacterial DNA is cut with, again, that more rounded shape, and the human DNA is cut with this much more jagged shape. Those pieces look like they're not really going to fit together too well, right? But if we cut both the human and bacterial DNA with enzyme 2, and they each have the same shape, like in this bottom picture here, now they fit together really nicely like two puzzle pieces. As long as we use the same of these enzymes to cut two different pieces of DNA, both types of DNA will have the same shape because they were cut with the same type of molecular scissors, and that'll let us put them together really, really effectively. And then we just basically have to take another enzyme and have them glue the whole thing together, tape it together, however you want to work that. But we're going to attach our DNA pieces, and now this bacteria can make human gene X really effectively. Here we have a little more detailed diagram of what we're looking at before. So on the left, we see a bacteria. And if you remember that from much earlier in the year, they don't have a lot of DNA, so it's small in a circular shape called a plasmid. On the right is some kind of animal cell, and we see one section of its really large DNA molecule. We wouldn't be able to see the entire thing if we see one small piece of it. If we cut each of these DNA molecules with the same enzyme, the cut DNA pieces will have the same shape, like we saw with the fancy scissors, so we can fit them together nicely, and when everything is fully attached, we have a bacterial DNA plasmid, a little round piece, that now contains some kind of animal gene. So right here, we took the animal gene out of the animal cell, put it into the bacteria, and now we have this little modified plasmid at the bottom. Once this plasmid gets put back into bacteria, that bacteria will be able to make whatever the animal DNA is instructions for, and that's the idea behind recombinant DNA. Anyone know what cloning is? What's a clone? According to our friends at Merriam-Webster, clones are identical copies. So they use a lot more words to describe it at the bottom there, but that's really just what this definition here amounts to. Identical copies of something with the same DNA. They have the same DNA, so they get the same instructions. They're identical. And look, they actually use that term recombinant DNA at the bottom. That's encouraging to see. Somebody at Webster's knows they're talking about that. That's always good. So good job, Webster's. So technically, identical twins are clones, so they have identical DNA. And there are plenty of other examples in nature, this kind of thing, where something copies itself, or there are multiple versions of something with the exact same DNA, and they just kind of reproduce like that. So this is really not anything unusual. twins are obviously very natural and common. However, you probably hear this word clone more often in the other usage, which is a verb, in where you're actually making a copy of something. It's not just that there is an identical copy, it's to actually make the copy. And that's, really where it gets kind of boring. It can get very interesting scientifically, but not probably the kind of thing we really want to see a lot of in the real world, because how unexciting is this picture down here with everybody just looking exactly the same? But let's see how this would work. This diagram here is how cloning works. The first cloned animal that at least anyone publicly is able to admit to was a sheep named Dolly. So usually you see these sheep in the diagrams and any pictures you may see of this kind of stuff. And in this one we see sheep A up at the top here and sheep B down at the bottom. We're going to use these two animals as a starting point to see how cloning actually is going to take place. So we're going to start by taking a body cell from sheep A, which has a complete set of DNA. So that's right here. Some kind of body cell. Now this could be a skin cell, muscle cell, whatever. As long as it's got a complete set of DNA and we can do good work with it. It has a complete set of the instructions for making one of sheep A. Every instruction for making a sheep A is in one of those body cells. And we're also at the same time going to take an egg cell out of sheep B at the bottom. So we take an egg cell from sheep B and a body cell from sheep A. This egg cell has some DNA from sheep B, so we'll need to remove it by removing the nucleus. So that's what you see here. They just actually take the nucleus out of this cell, and right now at this point you basically just have like a little kind of empty sphere. It's some of the cytoplasm and the organelles, but doesn't have any DNA in it, so it cannot grow into something new, which is basically an empty egg. So once that's done, we have this hollowed out egg, no nucleus or DNA inside. At this point now, we can combine everything. So the DNA from sheep A is put into sheep B. So we'll take the DNA out of this egg out of this body cell up here, and just actually install it into that now empty egg cell at the bottom. And at this point, the whole thing will start to develop into a new organism. So the cells will start to divide as they would normally after fertilization, and start to grow into these little kind of balls of cells up here. And once this is going on, we can implant this whole embryo, this whole little bundle of cells, into a new surrogate mother, sheep C. So we now have a third animal, and we're going to implant or install this little bundle of cells there that started to develop into a new baby sheep into this animal C, and just let it go through the whole pregnancy and development as it normally would. When sheep C now finally gives birth to a baby calf, or baby sheep rather, it's now going to be an identical clone, identical copy of the original animal A up there, because that's the only source of DNA. We took all the DNA out of the cell from sheep B. We took no DNA from sheep C. It really is just kind of there as a artificial mother, but all the DNA in this new little lamb down here is identical to sheep A, because that's the only source of DNA for the animal. So that's how we get an identical clone or a copy of sheep A, our original animal, which is not at all related in any way to the other two animals, B or C. So we're not quite to this point just yet as a society, but who thinks this is not an inevitability? Coming soon to ESPN 7. So that's the end of biotechnology for now. We're going to move right along, as I said before, and get into lesson 5, which is called gel electrophoresis. So we're going to move right through this because it's not that much. It'll be, again, a little bit of a longer video today, but then we don't have anything new to do Thursday. We'll do that cool lab that's online. So looking at our questions here, why do we want to tell the difference between DNA sequences? Well, in our world nowadays, we usually see this kind of thing with maybe different law enforcement practices, seeing if someone committed a crime. You may see the kind of thing that possibly is on some kind of trashy talk show or something like that with, who's related to whom, or is this person a relative, anything like that. And as for how we can actually identify the different DNA sequences, we're going to see now how we can use the techniques we just saw with the different enzymes that can cut DNA up, and how that's actually going to be applied by us in a either commercial way or a legal way or some kind of industry. So some interesting stuff, and we'll see how this goes. We just looked at how different restriction enzymes cut DNA in different ways, depending on the exact order basis we're looking for. Now we'll see how we can use that to our benefit. Since everyone has different DNA, the same restriction enzyme, that same thing that's going to cut the DNA, will cut everyone's DNA differently. Our DNAs have different sequences, so it's going to cut it in different places. So that means if we take samples of my DNA and your DNA, and we cut both of them with the same restriction enzyme, look what's probably going to happen here. Because we have different DNA sequences with different letters in different orders and different places, that one enzyme will cut my DNA in 6 places, potentially, and maybe only cut yours in 5 different places. And it sure looks like it really only has one spot right here where they happen to cut it, just kind of dumb luck, in the exact same place. So we shouldn't expect that's going to match up really at all. The important thing to notice here also is that some of the DNA pieces are really, really small that are made. So like on mine, if it cuts them really close together right here, or on yours down here, they're pretty small pieces. We get these really tiny pieces of DNA, whereas others are huge. I mean, look at this on your DNA down at the bottom here. This is a very large piece of DNA that's left over between the two cut sites. And then we have multiple other kind of middle sizes in between. So these different sizes pieces of DNA, just made by exposing these different DNA sequences to the same enzyme, give us a practical way to compare DNA, which is commonly referred to as a DNA fingerprint, a phrase you probably heard at some point over the years if you've ever watched any kind of CSI or Law and Order or show like that in the last 20 years or so. And at its core, it's really no different than taking a group picture and making sure taller people stay in the back and smaller people up front, which is going to give us a way to compare things based on size. DNA fingerprinting has a variety of uses, mostly to help in criminal or civil investigations or to establish whether people are related to each other. We often see pictures that look kind of like this. So each of these columns is a different person's DNA, and all of these little lines that we see is a different piece of DNA, with each one indicating a different size piece, like we saw in the last slide. Everyone's DNA is different, but the more similar two sequences are, the closer those people are related. And keep in mind that since everyone gets their genes from their parents, no one has a truly original DNA sequence. So we all have a mix of some combination of our mom's genes and some of our dad's genes. So in that picture at the bottom, each one of those little colored lines you see is a different size piece of DNA, just like we saw before. We cut the DNA up into little multiple size pieces with different enzyme. Remember in lab we tried figuring out who made everyone fake sick with that lab with the cups? We had to go off of patterns to trace the spread of disease, but in real cases we can easily test DNA when we need to. As I'm writing this, we've already had multiple New York labs use genetic testing to trace coronavirus from New York back to Europe, not from Asia as we originally thought happened, and what actually does seem to have happened on the West Coast. We can also tell that the virus originated in bats and was in this country for months before anyone even knew it. Now, we don't really need to do much detective work in this case because we can just go to DNA. We don't have to rely on guesses or hunches because DNA will tell us the story if we let it. Let's cut some more DNA like we did a couple minutes ago. The SMA1 enzyme here is one we haven't seen before, and its recognition sequence is going to be 3 C's followed by 3 G's. And then when you find those 6 letters, you're going to cut right between the first and second C. So let's try to find that sequence in this piece of DNA right here. So look for 3 C's and 3 G's. So I'm going to start on the left and 1, 2, 3, 1 right there. Okay, good. 3 C's and 3 G's. And now I'm going to cut between the first and second C. So I'm going to slice it right there. And let's see, going on. 3 C's and 3 G's. Here's 1, 2, 3 C's, 1, 2. Yep, another one. 1, 2, 3 C's, 3 G's. So again, between the first and second C, I'll cut it. And let's see, any more on here? C's and G's. 1, 2. Is that the one at the end? 1, 2, 3 C's, 1, 2, 3. Yep. 3 C's and 3 G's. So I can cut one more time at the end. Okay, so we cut it 3 times. And that's going to be some of the answer to our questions here. We cut 3 times, getting 4 different pieces. So we got 3 cuts, 4 pieces of DNA. Our questions ask, how are these pieces different? Well, they're different sizes, right? This one seems to be pretty big right here. The other 3 seem to be right about the same size, maybe identical. Maybe they're off by a letter or 2. But we have 1 larger piece and then 3 that are a little bit smaller. In terms of how we can sort these pieces, that's what gel electrophoresis is. That's the topic of this last lesson in our chapter, and that's where our lab's going to be on Thursday, how we can actually sort these pieces of DNA now that we've randomly cut them with whatever enzyme we choose to use. And that's going to be what our fingerprint is. We can sort DNA, separating a wide range of pieces of varied sizes using a process called gel electrophoresis. And this uses electricity to spread the pieces apart from each other. So you kind of charge them up and then use this electrical field to make them actually push apart and separate. First, we use an enzyme to chop up DNA into all those little different sized pieces we saw before, which we usually call fragments. Then we take those fragments and inject them into a block of a gel that's called agarose. So this is kind of like a very dense piece of jello that's just shaped like a thin rectangle. So you can see there it's a rectangular shape. That's looking at it from above, but it would be only about a centimeter or so thick. It's not that big. It's like very smooth jello, but again, very dense, really hard packed in. So on a microscopic level, inside the agarose, it looks a lot like this. Even though it's kind of smooth outside, if we could see it on a microscopic level, it's got all these little nooks and crannies and these spaces that the DNA can actually move through. So I always try to compare this stuff to Jello jigglers, and I always find then that no one knows what Jello jigglers are, unfortunately, because I guess the world has changed for some reason. I don't know why it's not a thing anymore. So to try to help you understand what I'm talking about here, here's a commercial from 1991.
We're here to celebrate a different way to eat Jell-O gelatin. With our bare hands. All we need, cookie cutters, this easy recipe, and we've got jigglers!
Jiggle, 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 those jigglers. Jiggle, 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 those jigglers.
The recipe's on the box. Jigglers, they're a handful of fun. Hey, put that back.
J-E-L-L-O.
That commercial has really not aged well in 29 years, has it? In any case, jigglers were fun and delicious, and all you really had to do was just use twice as much Jell-O powder or less water than normal, so they came out firmer and thicker than typically jiggly Jell-O. But back to science. Once we have our gel slab done, we put it in a special box and pour a liquid called a buffer over, and that's really just this salt liquid that helps things move. So we put in the box here, and then we plug the entire box into a power source. Now, at the top of the gel are these little holes called wells. You see right here. They're just little indentations. So like you see any kind of well in the ground. And that's where we actually inject the DNA. So we put the DNA into those little wells there. And then once you plug it in, the end near the DNA gets a negative charge. So this little electrical terminal right here, that gives it a negative charge. And the other end down on this end of the gel gets a positive charge. The trick to this is that DNA naturally has its own bit of a negative charge. So since similar charges repel and opposites attract, the negative DNA that we just loaded into these wells right here, start to move down in this direction. They start to get pushed away from this right here, the negative side, and get pushed towards or pulled towards the positive side down there. So the DNA actually starts to move. And that's where you see the little lines here. That's the DNA moving through the gels in their little fragments. So they start to move down to the opposite side from where they started. At this point, we call this migrating or migration, like you have people or animals migrating too. So remember how the inside of the gel has a little kind of web-like spaces to it? The smaller DNA fragments are able to squeeze through these spaces a lot easier and move a lot faster than the bigger pieces, like these mice running through forests there. The bigger DNA fragments are like the elephants, and they keep getting kind of stuck in the spaces, and they move a lot slower through the gel. They're going to get through, but it's going to take a lot more time. So they don't move as far in the same amount of time. So since DNA usually just really doesn't move that fast in general, we usually will turn this box on, let the electricity push and pull these things, and let it go for, typically a half hour, 40 minutes, somewhere in that range. And at the end of that time, the bigger pieces are still pretty close to where they started. They haven't gone that far, whereas the smaller pieces get pretty close to the other end typically. And you can actually use a dye. In this picture up at the top there, you see these kind of like little purplish, reddish lines there. You usually would put some kind of dye in with your DNA sample so you can actually see how far it's gone. And just you'd stop it somewhere right around this point. By the time it gets mostly to the other end, you're usually pretty confident that it's gone far enough. And then you turn the electricity off and the DNA stops right where it is. So you can see here how this works. The smaller fragments of DNA end up somewhere down here. And they kind of start in the middle. They'll end up somewhere near the bottom part here. And then we see this line kind of here on the gel. While the bigger pieces only move so far and get somewhere mostly up near by the top here, somewhere maybe in the middle eventually. When we look at the gel, we don't actually see pieces of DNA. So we look with our naked eye, we're only going to see these banding patterns. We'll see this kind of straight line across or a little smaller ones because these are microscopic things. We just can't see it. So we'll see the bands, and we can usually, like I said, make them glow with some kind of chemical, put some kind of loading dye in. Sometimes you'll put a UV light, and it'll make it light up in different ways. But usually it's pretty easy to illuminate and then actually see where our DNA is, and that's what this would be now on the bottom picture. So it's easy at that point then to look at the gels and compare samples. So in this one at the bottom, we see 4 different samples. So you've got people or individuals 1, 2, 3, 4, and maybe sample 1 is a blood sample from a crime scene. The other 3 are suspects. So we've got a blood sample with DNA that we know or think is a person who committed a crime, and we want to figure out who matches up those samples. It's a little bit like fingerprinting, except it's being a lot more specific. So whose DNA seems to match sample 1? Well, if I'm looking at this, it looks sure like 1 and 2 are identical, right? So that would be a pretty good indicator at that point that individual 2, suspect 2, is someone you're going to look at. But this is going to be a lot more specific, like I said, than fingerprinting or even blood typing. Blood typing can tell you is someone type A, type B, type AB, whatever. But DNA is specific to a person. This is going to give you really, really, really close match specific results. Whereas, say, like sample 4 here, that doesn't look anything like sample 1. So sample 4, person 4, would be eliminated pretty quickly from consideration by the police, hopefully. So just remember this point. Bigger pieces of DNA stay up top on the gel. They stay closer where they started. You can see again over here, all these bigger samples don't really move that far, where the little pieces, they go pretty far and move a lot more down towards the other end. One last note before we finish this. A benefit of having the DNA kind of stained and colored with this dye is that you always know where it is and can turn off the power source in time. So I said we usually let it go for about a half hour or so. That's just a rough estimate. Sometimes you can turn the power on, have it go a lot quicker. You can have it go slower, and it'll take a little bit longer. You may get some better spacing in between these things. But the risk there is that if you leave the electricity on too long, the DNA will actually move straight out of the gel. It'll just keep going through these spaces and just go straight off the edge here, like when people thought the Earth was flat and you could actually sail off the edge. And at that point, the DNA is just gone. I mean, it just gets pulled straight out of the gel. It'll go into the liquid, the buffer surrounding it, and it's lost. You're not getting it back at that point. So that'd be kind of a wasted experiment. You wouldn't get good results. So it's important to be able to know where this stuff is so you can say, uh-oh, I better turn this off here before my experiment gets ruined. That's all the new materials. Let's just do some practice questions to finish this out. We've got a picture of a gel here. We'll compare and just see what the question says and figure out what we're doing here. It says, as part of a laboratory technique, DNA samples taken from 4 plants are separated. So that's our 4 lanes, our 4 samples, and the results represent the diagram. So let's see. They start up here in the wells. This is where you would have injected the DNA and just kind of drop it in there. We'll see this in the lab in a couple of days. And then when the power turns on, it'll go in this direction. And it's spread out to make this banding pattern. We're going to see if we can compare things. So it says, circle the band in the diagram below in the results for plant X that would contain the smallest DNA fragments. Well, we know the bigger pieces stay up here because they just don't move that fast. If we turn everything on and let it go for about a half hour, the bigger pieces are going to stay down up there, and these smaller pieces are going to move a lot farther in that same time frame. So your smallest pieces, it says the smallest DNA in plant X, that's always going to be right at the bottom, so that's your smallest piece right there. So it just gets smaller as it goes down. Your smallest piece is right there. The smallest pieces in the other ones are also the ones that are farther down there. Lieutenant Russ is investigating a murder scene. The felon was scratched by his victim and some of his skin cells were found under the victim's fingernails. A DNA test was performed, and which of the suspects is the murderer? So just looking at this, it hopefully jumps out pretty quickly which one is going to match up. But you're looking at the felon right here where you know this is the DNA sample from the person who committed the crime. Which one of these things looks like that person? You can eyeball this. It's not a lot to really compare. If you really want to get detailed, you say like suspect 1 has two little bands right here, and they are not in the felon column. They're missing there, so that's definitely going to eliminate that person. There's a lot of other things like this band right here is missing. This one here is missing. If it's not matching the felon, this person doesn't match, and just easily we hopefully can see that suspect 2 is identical to the felon. All the DNA is the same, so that's going to make them the suspect that the police want to pay attention to. I saved this question for last because this is a little bit trickier, not really, but just illustrates a really important thing you need to remember here. I mentioned before, all of our DNA comes from either mom or dad. So we have this question here. It says, Mrs. Smith has a baby named Tyra, and she believes that one of 2 men can be the father of her child. A paternity test is done. The results are shown above. Which of the 2 men are baby Tyra's father? So we have the baby's DNA right here, and we have mom's DNA. Again, going back to the idea that the baby's DNA had to come from either mom or dad, we should not expect her to have the same DNA as either parent. It's going to be a combination. So as long as we know where the baby's DNA came from, we can figure out who the parents are. So we want to start with the baby. That's the one we care about. And I'm going to look at this line right here at the top and see that it compares very favorably to mom. So she got that gene from mom. The next one down, the baby, mom does not have that gene. That means that DNA had to have come from dad instead. So I'm going to leave that blank for now. I'm going to go through now and just match up where the baby got the genes from her mother, and then anything that's not accounted for mom had to have come from the dad. So this sure looks like it came from mom because they seem to match up. The next one down, this one does not match the mom, so we got to look for that. I'm going to go back actually and circle those ones in a minute. This one seems like it came from mom, and the one at the bottom looks like it came from mom. They're a little bit offset, it seems, a little bit, maybe not perfectly lined up, but they're close enough where it looks like that's where mom's DNA was found. So going back, the baby had this banding pattern right here that does not come from mom. This did not come from mom because it doesn't match anything there. This did not come from mom. It doesn't match. This did not come from mom. It doesn't match. So we now just have to find a dad's DNA sample that matches those bands, and that should tell us who the father is. And remember, all the DNA has to be there. You can't have dad having genes that shouldn't show up, or the baby has to at least come from dad. So right here, if we go through, it's pretty close to dad 1, but it sure looks like it's going to match dad 2 a little bit better. For this band right here, could be dad 1 potentially. It could also be dad 2. They're kind of close. If I follow this third pattern around, I see nothing for dad 1, but it looks like it's going to be matching dad 2. And same thing for this one. There's nothing there where we see dad 1's DNA, but dad 2 has all of these bands. So it looks like dad 2 is going to be the father of this baby. So just back to what I was saying a minute ago, if we look at where I'm circling in green now, dad has these genes here, which don't match up in the baby, but that's fine. You know, he didn't give all of his DNA to the baby. He only gave some of his DNA to the baby. Mom gave other genes. You don't have to have the parents exactly matching the kid, but the kid's DNA always has to be accounted for by some combination of the parents. That's a really, really important thing to keep in mind. And with that, we are done with genetics. Next up, we're going to do a virtual gel electrophoresis lab that I'll put probably on Thursday. I'll put it into OneNote. I'm going to give you probably a 2 or 3 minute video just introing everything because there's some issues with the website that we're going to use. But you'll see where everything is. It'll be a nice little simulation. You'll actually make the gel, you'll load the DNA, and see how that whole thing works when you run and compare samples. That's it for now. So go finish the form for today in OneNote. Put some worksheets also up in OneNote. I will have extra help on Friday at 12 noon. That's, I forget what the date is. The 1st, I guess, actually. 1st of May, right? The work from this week, so the things I'm giving you for today, the forms, the worksheets, everything is due next Monday night. As I said before, we will have a virtual lab this week. So that's going to be Thursday. It's Monday when I'm just finishing up this recording. I may very well just put everything up for you on Tuesday. I'm not sure if I'm going to give you all in one shot and then you've just got the whole week to do whenever you need to, or if I'll wait till Thursday to post it. But in any case, you'll have the virtual lab at least on Thursday for the gel electrophoresis. And then Friday, again, I'll have extra help if anybody has questions with that or anything else. Additionally, next Monday, I'll have extra help again. So reach out with any questions. Keep in touch. Look out for each other. Have a good weekend if I don't talk to you until then. And I'll see you at some point.